And I want to really talk in my 20 minutes about what we've been doing, my work and the work of EcoHealth Alliance and others, and trying to understand emerging diseases and trying to predict them. And really over the last 20 plus years of trying to get a handle on how often they emerge, why they emerge, and can we predict that process? So of course, this is a very important issue, and not least of which because emerging diseases are increasing over time. We've got data on that, we've published it, corrected for reporting bias, which of course is increasing even more over time. But you can predict pretty safely there will be about 5 new emerging disease events. Some of them will be very minor, a few people infected. Some of them could be a pandemic. And in fact, all of them could be a pandemic. So that's one of the key issues. To really predict pandemics like COVID, we have to try and get a handle on each of these small events and what makes them emerge and then what makes some of them become pandemic. And the other big issue is the economic cost. Pandemics like COVID cause us a lot of mortality and horrific impact on our personal lives, our relatives who we lose and our loved ones who we lose, but also on a global scale, the economic impact is truly a shock to the system. And this is just a rough estimate of some of the outbreaks over the past 20 years or so. But if we think about COVID, we now have this looming black horror on the side of an estimated cost of more than $16 trillion by autumn this year, fall. Really quite incredible. And the reason why this cost is so high is because we rely so significantly on a globalized economy. And really, pandemics and emerging diseases, there is good science to show that they are a product of our globalized economy. They're a product of our exponential growth in population, in exploitation of the environment that drive us into close contact with wildlife, where the majority of pandemic pathogens originate. So there are many things that are positive about our globalized economies and our living through the Anthropocene and this great acceleration since the 1950s. And you can see telecommunications at the bottom, just incredible, and how our lives have been changed by that. But there are also many significant negative impacts, including CO2 pollution, over harvesting of fish, for instance, quite dramatic, and tropical forest loss, increase in land use for domesticated livestock production. These are the things that drive pandemics at a global scale. What we now need to think about is what can we predict about that process. So I'm going to start by showing what we can do, what we have been able to do, and then pose a few key issues of the things we can't do and what we need to really try and get a handle on. So there's a sort of schematic that Chris Crowder-Johnson showed for how pandemics emerge. And really what happens, if you look at the bottom of this slide, we've got animal populations that have a natural assemblage of pathogens. And I'm going to talk mainly about viruses for the rest of this talk, because viruses tend to be the highest impact emerging diseases on people. So we have animals like bats, rodents, primates, and others that carry a huge diversity of viruses, much of which is unknown to us. We move livestock into their areas, we build roads into the forest, we hunt them and eat them and trade them. And those pathogens make contact with us, either through livestock or directly. And then they either take off or they don't, because they're able to infect us and spread or not, or they evolve that capacity. And what we see as an outcome are what in the middle, this sort of stage 2, local outbreaks of new diseases. And we see these quite regularly around the planet, and many of these probably go undiagnosed, just like the early stages of COVID were not detected straight away. And they often mimic other diseases, just like COVID mimics other pneumonias. And eventually we find out the subtle differences, but at the beginning it's hard to see that. So good examples are Ebola, where we've seen repeated outbreaks of 400 people or so infected in villages in rural Africa. But then every now and again, a more explosive outbreak when the pathogen spreads through a dense population of people in more urban areas. So understanding that rural to urban connectivity is going to be critical for prediction. Understanding which pathogens can break out and which can't is going to be absolutely critical. And then some become pandemic. And I think that that's probably the most easiest to predict. Once pathogens have the capacity to spread human to human, people get on planes and travel, and the pandemic begins. And we've been tracking air travel patterns and predicting with algorithms where people will change at hubs to get other flights. And you can get some pretty accurate predictions of where a disease will spread to once it emerges. And that can be used effectively to increase quarantine or control measures. But all of the other sides of this are very difficult, and persuading people to use those to actually create policies is even harder. And I want to talk a bit about that in a minute. So what have we been able to do over the past 20 years or so? I think we've done a lot. We've got the first steps of what I consider prediction of pandemics. And that's a loaded phrase because, of course, we don't really predict pandemics. We know they're coming. We know roughly where. We know what species. We know what types of viruses. But predicting exactly where or when is very, very difficult, a bit like predicting earthquakes. But we can tell pretty accurately where these sort of hotspots for emerging diseases lie. And we've done this by tracking prior emerging disease events, correcting in our analyses for where people are doing the most surveillance to try and find the under-surveilled places on the planet. And then looking at correlations between where we think emerging diseases begin and what's there driving them. And what we see are very crude, large spatial scale projections of risk. And this is a risk map. It's an emerging disease hotspot map. It's a projection of where on the planet likely the next pandemic will originate. It's a measure of human population density, where people are on the planet, where people are coming into contact with wildlife diversity and the diversity of their pathogens, and especially where there's a change in population or a change in land use, and an environmental expansion that pushes people into contact with wildlife. But it's very crude. And even though we can look at these correlations in a sort of global grid square on the x-axis by grid square, and on the y-axis the risk of a relative risk of an emerging disease event, we can see how biodiversity tracks with pandemic risk. We can look at how in the middle here on the bottom right of the screen, where there's zero change over the past 10 years in forest cover or pasture cover, there's a very low risk of emerging disease. And where we increase forest or decrease forest, we get an increased risk of emerging diseases. But again, these are large scale patterns and quite crude. So to actually go to a local region and say, here's the risk, it's coming in 5 years, is not yet possible. And that's where we need to go. In terms of which animals we should be looking at, we've got good data on this. This is work from Kevin Olivelle, published in Nature a couple of years ago, looking at every species known to carry viruses, including people, mammal species, and looking at viral richness, the number of species that each group of animals carries. Each dot on this plot is a species. The red dots are domesticated, the gray dots are wildlife. And of course, domesticated species have been worked on a lot more, so we see a lot more viruses. When you look at the proportion of viruses that are zoonotic, able to infect people, we see a shift, and we start to see primates, rodents, carnivores, and bats light up as the key risk. But modeling this and correcting really for the underlying biases in surveillance, we really do see some groups jump out as a key risk factor. So bats in particular have the highest proportion of predicted zoonotic viruses of any mammalian group. And that's significant. So if we're going to look for the next pathogens, we really should be looking in the groups of mammals that likely carry the zoonotic viruses that we don't yet know about. Using the residuals of these correlations, you can come up with, and by looking at the most studied wild animal, and then saying, if we studied all wild animals at that same level, what would the number of previously unknown viruses be, and which can become zoonotic? We end up with an ability to produce a map of where the risk is in terms of unknown viruses. So if we want to do viral discovery programs, these are the places we should go to find the highest number of potential zoonoses. Now, some of these places are emerging disease hotspots. Others are very remote rural areas with little human activity, so little chance of a disease emerging. So you've got to bear that in mind as you work through these programs. And this problem with all of these analyses are the data that underlie them. And I think that's one thing that we've found over the past 20 years. The data are not there for many of these things, including viral diversity. We're looking at a fraction of the known, of the unknowns in the knowns in the viruses. We're looking at a fraction of the number of outbreaks out there, most of which probably go undiagnosed, and small clusters of cases in rural areas where clinics are hard to get to or people are too poor to go to them. So there's a lot that needs to be done. And I often use this quote. Donald Rumsfeld was exactly spot on with this quote, and he did win a Foot in the Mouth award from a journalist society for this, but actually it's very prescient in terms of both terrorism, which it was designed for, and viral diversity. There are efforts on the way to better describe wildlife viruses so we can find out what's out there. And I think as you develop your clever algorithms and cool machine learning tools and sensors, it will be really important to tie in with groups that are out there doing this work on the ground. So the very latest field data goes right into your algorithms, and you can use that to really improve predictions. And that's where I see a real way forward. We tried to do this with the Predict project from USAID, and we're quite successful. These are coronaviruses from bats. And what you can see is the more samples from bat species that you test, the more viruses you discover. But that discovery curve starts to plateau. And using algorithms from conservation biology for protected areas and the diversity of the number of wildlife in a protected area, you can predict to some extent the likely unknown diversity of viruses in these groups. If you then extrapolate, and by the way, all of this work will require extrapolation. It will require creativity in finding proxies for data that don't exist, and really clever ways around that. And that's where the creativity will come in. And I do think that, mixing engineers, biologists, anthropologists, social scientists, veterinarians, and medics is the way to do this with that level of creativity. And we can come up with a very rough estimate of the number of unknown viruses on the planet. In mammals, which are the highest risk for emerging diseases, we estimate around 1.7 million unknown viruses. Now, that may be an overestimate, it may be an underestimate. We need to go out and check. We need to go out and do the background work to find that out. But there's one thing for sure, this is an achievable number of unknown viruses. We could go out and discover these for a few $100 million a year. And that is the level of expenditure on other programs to deal with pandemics. And it's a fraction of the cost of just this one pandemic alone, $16 trillion for COVID. So we proposed the Global Virome Project. Others in the op-ed in New York Times yesterday proposed a sort of forecasting strategy based on sampling people for the first signs of outbreaks. There are many, many other efforts out there. These will start and continue, link in with them, use the algorithms to work with those data, and we will massively improve our predictive skills. So what are the key questions, and what can we start to think and do about them? Well, one thing that's clear, as we find new viruses, and we found a lot of them in our work and work with others, it's not possible just from a sequence yet to predict if that virus is going to be even able to infect people or become pathogenic or become a pandemic. So that's a big black box that needs to be looked at. And there's some real interesting work to be done there in terms of looking at sequences and looking at viral diversity. But there's also interesting work to be done in all the other factors too. And this is a paper that's going to be out in PNAS soon from Zoe Grange and others at UC Davis and part of the Predict team. I'm on this too, that tries to rank the other factors, the ecological factors, the viral trait factors, the host trait factors that predict the capacity of viruses to become pandemic. And to say, when we find a new sequence of a new virus, can we rank it and say it's a high risk or a low risk? Again, it's a crude estimate, But it has some use, because then you can decide whether to spend money, sequence the full genome, and actually do some experiments in the lab to see if it could infect human cells, to see if it could cause disease. We've done this with SARS-related coronaviruses over the past 15 years, I think quite successfully. We were looking for SARS-related coronaviruses in China. We found a huge number of them. These are from bats. You can see SARS-CoV-2 there as well, and the pangolin viruses and the closest relatives of SARS-CoV-2. What we were able to do is to say, well, here's a genome of a virus that seems to have a receptor binding domain, a spike protein that could bind to human cell receptors for SARS. And then using cell culture and mice that express human ACE2, which is the receptor for SARS, you can then demonstrate whether that virus can infect human cells and mice and even cause SARS-like clinical signs in this mouse model. So there is a strategy there. It's expensive work to do, but it can then fine-tune the predictions from your work as you start to look at viral sequences and do the AI work on those genetic sequences. But there's a lot more to do. So the other big issue, and of course, I appreciate having an anthropologist on the talk, as well as the coolest background on Zoom today. Also, this idea of, this is a human issue, and humans added an incredible amount of complexity to this that's, again, hard to predict. So what are we going to do? Can we pinpoint the interfaces that are going to be the highest risk? Can we pinpoint where and these environmental changes are going to shift wildlife populations and drive some signal that we can spot? And can we map human behavior risk? Now, we've started to try and do this, and it's not easy. First of all, you need big experiments on the ground. Studies, I should say. You need to work in areas that include urban centers, rural areas, and even pristine forest areas where wildlife are more diverse. If you're really going to understand the process, because you're trying to recreate this process of land use change that leads to emerging diseases in this study. And so here we looked at 3 countries in tropical forest regions with high diversity of wildlife and human activity. We selected low disturbance areas, pristine forest, intermediate disturbance and urban areas, high disturbance. And we sampled wildlife in a standardized way, bats, rodents, primates. We tested them for a known group of viruses using the same primer sets for PCR. And we did viral discovery on those, and we looked for known and new agents that have the potential to be zoonotic.
Peter, we have two more minutes, please. Thank you.
We then did standardized surveys of people, but it's very, very complicated. And what we find is intermediate disturbance areas have a highest risk for viral spillover. So I'm going to skip to the end and talk about the sort of work we will need to do, and I think is very important, as well as tracking the rural origins of viruses here in South China into the wildlife markets, measuring those pathways. This isn't just about road networks or airline traffic, it's about human activity along those pathways. And then finally, how do we get governments to work together to actually use the data and use the analyses to do some prevention and control? And what we've tried to do is focus on land use change and talk to governments about the cost, both the profits from those activities, which obviously drive them, and then the potential cost, measuring the number of diseases that could emerge for every hectare of land. And what we do see, and looking at COVID in particular, is zoonoses costing about $1 trillion a year to the planet, whereas strategies to reduce deforestation and wildlife trade are about 2 orders of magnitude less than the damage produced. Obviously, we now need to do more work on how those two connect and how much reduction you get for each hectare of forest saved. So there's a huge amount to do on that that's going to be very profitable, but will actually help governments make decisions